Yeah, so I think we have some issue in our HPLC. I think mixing is not happening properly. Everything is eluting at the end. So I had to call the engineer. So at that moment, like our system, this system since beginning 1 year, CMC was there, warranty. After that, I have not taken any CMC or MC. It was running last 6 years without much problem.
A very good afternoon, everyone. I welcome all of you for today's Science Setu lecture series. This series is organized by RCB to commemorate 75 years of India's independence and also to showcase the science that we do in RCB. And it is my pleasure to introduce today's speaker, Dr. Tushar Maiti, for today's talk. Dr. Maiti earned his PhD degree from Indian Institute of Technology in 2005 and then gained postdoctoral experience from Nagoya Institute of Technology, Purdue University, and Wisman Institute of Science, after which he returned to India and joined Regional Center for Biotechnology. Currently, he's working as associate professor at RCB, and his group is working towards understanding the mechanism of protein misfolding, protein overload, and cellular dysfunction. The other important project that involves identification of biomarkers which are relevant for early prediction of protein. Now, it is very much evident from his publications that are published in various internationally reputed journals. So with that brief introduction, I would like to request our speaker, Dr. Tushar, to deliver a lecture on the topic Parkinson's disease pathology. Over to you.
Yeah. Thanks, Nidhi. I think but she gave long introductions. So anyway, it is for me. It is basically pleasure to give lectures in front of our students. Generally, we every year we deliver 15-minute lectures in front of all the audience during our program advisory committee meeting. But today I will get at least a little bit more time to discuss whatever we have done last few years. So as Nidhi suggested, today I will be talking about the Parkinson disease to the clinical, basically illustration of the pathology. However, the molecular understanding and its complexity associated with different molecules which drive the disease pathology. So before moving into this particular topic, I will just introduce my it is hanged. And oh yeah. 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 So the people, those who are involved, this is the important slide, those who are involved in the study of different aspects, as Nidhi mentioned. We study the Parkinson disease related work, as well as the large number of people are working in the biomarker discovery, child health. So today, this is the present group member. Majority of them are basically involved in Pitambat project. Along with that, some people are in the Parkinson disease related work. So today I will be talking about some work related to Dr. Monisha, who recently graduated, Roshan, who was my second student, Sanjay, and Rinki. So some of the work I will discuss today's lectures. So Parkinson disease, first of all, I think this component is very much aware of all the community here. These actually neurodegenerative diseases after like Alzheimer's, and like it actually described 1817 by James Parkinson initially as palsy, because it described the pathology, or like you can say phenotype, particularly the shaking disorders, and it is related to the chronic progressive disorder occurring in the central nervous system and degeneration of neurons producing cells in the specific to the substance nigra region. So midbrain, which substance nigra is part, where majorly the resident like neuron cell is dopaminergic neuron. So loss of dopaminergic neurons leads to the formation of different types of like physiological changes along with the different kinds of phenotypes. So clinical phenotypes. So here disease prevalence, if we 100 to 200 like numbers of individual among the 1 lakh of basically the populations, we see that like PD is basically estimated. Annual occurrence is approximately 20 per 1 lakh, and it is ranging in the progression of the age, and male to female ratio is 3:2. So if we see the global map, like what is alarming, there is growth of like the Parkinson disease. The country where is basically demonstrated as a red color, India is one of them, like where we see that growth is more than 100%. That means more and more like the progression, urbanizations and many other factors contribute to the progression of the disease, and we could see that rapid progression of the Parkinson disease. So while I am talking about the Parkinson disease, another term I will Parkinsonism. So Parkinsonism and Parkinson disease like actually comprise of, or like you can say segregate substantially. So Parkinsonism is a phenomena, or you can say conditions where you could have like the movement related disorder, which is basically Parkinson. Along with that, variety other clinical symptoms, like you have depressions, you have many metabolic disorders. Along with that, like you have certain kinds of disorders which is not related to the movement, particularly speech related disability and memory. So all these complex phenotypes basically consider as a Parkinsonism. So there is a very subtle barrier between these two and Parkinson. So while somebody will be studying the Parkinson with majorly the movement related disorders, where like you see that like the person is unable to move, or like the facing the difficulty in the movement. So here the pathology, if we see, this is basically PET image scan, where this particular region represents the dopaminergic cells, where deoxyglucose intake, particularly the active dopaminergic neurons, is like intact by this particular small metabolites, and shows as it is a like the fluorine 18 basically molecule. We could see that like the normal conditions, if fluorine intakes happens properly, particular metabolites intake happens properly, you could see that distinct visibility of substance nigra region. If degeneration happens or the death of dopaminergic neurons, you could see like the distributions of this particular metabolites throughout the other space, and you will not see nice distributions in the substance region. So even though these PET scans are used, but it is very difficult to discriminate between the Parkinsonism and the Parkinson disease. That means many other neurodegenerative diseases, the PET scan image also shows similar degenerative behavior. So in that case, it is very difficult to discriminate. So that complexity is coming here, the discriminations of Parkinson disease along with the other neurodegenerative disease. one of the important metabolites which utilized by dopaminergic neurons and like the functions, all these motor coordination functions. So these are the component like now we degenerative conditions, or you can disease conditions. Tremors, a shaking like behaviors where the physical movements person is unable to move, difficult in movement. Bradykinesia, that means persons basically shows the very slow movement. This may be to the stiffness of the body, hand as well as leg, and like different type of postures, basically movement. And another component is cognitive impairment, which lead to the dementia. So we generally see the dementia is one important component in Alzheimer's disease. However, Parkinson disease also there significant amount of cognitive impairment during the progression of the disease. So dementia is also part of the PD pathology or PD basically characteristics. So if we see the pathology, the most important component we observe, there is a depositions of proteinous aggregates, we call it Lewy body. So Lewy body type aggregates are present there. So here the pictures which described here, the depositions of like large number of proteinous aggregates, and that is we consider as a Lewy body. It is by Fenridge Lewy, so that's why after his name this component is given as a Lewy body. So so what is the importance of the Lewy body? Particular molecules basically drive large number of, as it is a composition of large number of protein aggregates, it actually brings many proteins, or you can say sequester many proteins in the particular positions or neuronal cells, which lead to the disease progressions, or you can say cause the death of like the active neuron. So here if we see the Lewy body component, different factors, like you can say mitochondrial dysfunctions leading to the formations of Lewy body, lysosomal impairment, functional impairments also contribute to the Lewy body, ubiquitin proteasome system impairments also produce the Lewy body, A beta or like the alpha synuclein, which is one of the major component in case of like the Parkinson disease, they also contribute the formation of Lewy body. And reactive oxygen species, impairment of redox imbalance also contribute the formation of Lewy body, as well as like in Alzheimer's disease, we observe the aggregations or sequestration of proteins called phosphorylation of hyperphosphorylation of tau. So hyperphosphorylation of tau lead to the formations of like the aggregate formations, and all these components we observe in case of like Lewy body compositions. And among the many other proteins what described here, the alpha synuclein is one of the major ingredient which actually drive the Lewy body characteristics or Lewy body like these functions. So if we see the molecular mechanism, it actually contributed by genetic factors, which is very less like the percentage, like approximately 15% of population shows the genetic basically Parkinson disease. However, majority of Parkinson associated with sporadic form. That means it is contributed by environmental conditions. So here we just put few important like the molecules or important conditions which contribute to the or progression of the Parkinson disease. One is pesticide exposure, metal ion exposure, and rural living. So rural living means like a lot of people use their like the smokes and different kinds of environmental exposures are there that lead to the progression of the Parkinson disease. So again, the genetic factors as well as environment factor crosstalk each other and regulates their functions, leading to the progression of the disease. So here you could see again, as I discussed earlier, mitochondrial dysfunctions, ubiquitin proteasome dysfunction, as well as large number of metabolites which is basically produced due to the dysfunctions, due to the aberrant functions of different genes, also modify the proteins, different type of functional proteins which drive the cellular functions, and alteration of that particular proteins lead to the cytoskeletal disruption, mitochondrial disruption, and it actually lead to the cellular toxicity and subsequently the disease. Recent, like last few years, the gut microbiota dysbiosis, like people demonstrated that due to the alterations of basically gut microbiota signatures or like the changes in the gut microbiota can also lead to the Parkinson disease. So the gut and the brain axis is very much well established, and these components play very crucial roles, like the dictating the brain pathology. Yeah, so I discussed about the different types of genes regulate in the Parkinson disease condition. I'll be talking about one term called synucleinopathy. So synucleinopathy is basically a conditions where group of neurodegenerative. So here the Parkinsonism is one of the important factors, not only the Parkinson. So synucleinopathy, where alpha-synuclein is one of the important regulator of Parkinson disease, they actually different times the insoluble aggregates, the different like the alteration of like or selective vulnerability populations of neuron, as well as the glial cells, which is basically involved in clearing of the brain or you can say brain resident macrophages, which actually clear different type of misfolded aggregates from the brain region. So alpha-synucleinopathy is basically a conditions where alpha-synuclein drive the pathology. So we'll be talking here majorly today's discussion restricting related to the synucleinopathies. So why we will be studying the synucleinopathy, or like we will be interested to looking at the synucleinopathies or alpha-synuclein related work. So if you see, alpha-synuclein is a very small, you can say the intrinsically disordered protein. The meaning of intrinsically disordered, generally protein in solution present as a folded form. It has different types of secondary structures. However, there are group of molecules or group of proteins which are conditions represent like a basically disorder, like after synthesis from ribosome, primary structures. So they do not have proper like the secondary structural element in the given conditions. However, like once the environment conditions imposed or cellular condition alters, they adopt different types of conformation. So because it is intrinsically disordered proteins, it can adopt large number of ensemble structures. So large number of structures, you can say ensembles, structural ensembles it can produce. And because of large number of structural ensembles, it makes the systems very much complex, because you do not have a specific structures for particular protein functions. So it has been observed that it exists in the monomeric forms as well as tetrameric forms isolated in the brain, like in the brain samples. And its mutations A18T, A29S, A30P, E64K, 53T, 50Q, and G51D. These are the polymorphism we observe, and they are basically linked to the association with the PD. So large number of population-based study has been carried out, and people have shown that this kind of mutations are observed and they link to the Parkinson disease. Like the A53T, the most studied like the Parkinson disease mutants, and many of them already been basically incorporated in the mice brain, like the mice basically transgenic model, and demonstrated that this kind of mutations produced like the Parkinsonism syndromes in the mice. Large study has been carried out like these 30, 46, 53, 50, and 50. Find like more than like the thousands of publications are there related to these mutations, their behaviors, how they aggregates, the progression of their mechanism of aggregations. So that are already been studied. So A18T and A29S, these two mutants basically discovered in I think 2014 or so. So, and reported first times in the populations, Jewish populations, and then like this functional property of this mutant has not been studied. So as here we describe that it is intrinsically disordered proteins, but it has three specific domains. One is N-terminal domain, NAC domains, and C-terminal domains. So this particular NAC domain, which basically contribute the formations of the fibrillar aggregates, N-terminal domains which bind with the membranes, and C-terminal domains, regulatory domains, which basically control as well as N-terminal domain. And these C-terminal domains also plays important role in different other basically cellular functions, particularly calcium transport and other mechanisms. So here I have basically put one image. You could see the physiological functions of alpha-synuclein. Mostly we discussed about, yes, alpha-synuclein is associated with PD. However, or Parkinsonism mechanism. However, we need to remember that this protein is ubiquitously present many other cells and mostly present in the neuronal cells. So it major role of the vesicular structures which transmit particularly the dopamine. That's why its abundance of like dopaminergic cells is very high. So it form this kind of vesicular structures, but gene duplications as well as lead to formations of like alter the membranes or alter membrane structures and lead to the disruptions of vesicular trafficking of the neurotransmitter. So here in normal conditions, it actually take the neurotransmitter and subsequently it basically through like the exocytosis mechanism. Okay, so here we showed structures which almost 15-17 years ago. So it demonstrated alpha-synucleins acquire the alpha helical structures in the presence of membrane. So it shows the alpha helical structures of N-terminal and C-terminal domains is completely like the disorder. So in presence of membrane, it actually produces the helical structures. However, progression, basically aggregation kinetics, we observe that this particular structure, the NAC domain, transfer into the beta seed structures. Here, as I mentioned, like the A53T and A29S, these two mutants reported in the literature in 2014. So we are curious to know that how these particular mutants behave and their comparison the existing mutants which is already present there. We could see these particular mutants present in the intermediate. I am not giving the detailed like the structural detailed information related to all the experiment, but this information is it actually impaired the membrane binding because these particular mutations present there in the membrane binding domains. So here helical domain, particularly this position A18T. So this position of 18 is very much critical because other two residues, like 17 as well as 19, both position basically the other residues are there, L residues are there. So if we replace that particular residue with A, like so 3 A presents or alanines are present there. If we replace the 3 alanine, the residue to this particular neighboring positions, we observe that the T positions, T18 positions is very much specific for membrane binding as well as progressions of the aggregation kinetics. So here we have shown the structural features of individual like the aggregates in individual, you can say fibrillar structures. So the important you could see here, the structures basically shows very much like interesting periodicity. Like periodicity represents like here one particular, it is basically a twisted structures. So twisting from one position to the other positions, they represent how much particular angstrom is or how much distance basically it maintained for each periodicity. This particular periodicity also dictate the toxicity of the particular species. So if we see, even though from gross the structural information you could find their is very much similar or look like determined or calculate the periodicity, you observe that all of them actually shows different like periodicity distributions, and that distributions is basically linked with the toxicity of the molecule. So like as we mentioned, the different mutants drive different periodicity of the fibrillar structures. So how this fibril structure formed? So here, like as you mentioned earlier, with membrane binding, it produced the alpha helical structures. It formed 310 helix, and then subsequently the 310 helix convert into the beta strand. Those who are structural biologists here, I do not have to describe the 310 helix, difference between the 310 helix as well as alpha helix. So alpha helix basically represent like the I+4. However, the distance between you can say the carbonyl atoms and nitrogen atoms, so it represents 3 plus I+4. However, 3 plus 10s actually represent I+3. So that means the tighter alpha helical structures. So this convert into the beta strands. So it is a spontaneous process where in alpha helical structure, and rapidly these alpha helical structures convert into the beta structures, which is thermodynamically more stable. And if we basically conversion of a one molecule, alpha helical structure, beta strand conversion. However, if we consider the aggregation kinetics, where the monomeric molecules, they assemble together, attach to each other, or like you can say self-assembled formations produce the oligomeric structures. These oligomeric distributions ranging from like 6 to 8 or 10 residue, with time they actually form a large. Subsequently, they produced the structures called protofibrils, and these protofibrils, the mechanism where the elongation of this particular fibril, protofibril, happens in you can say longitudinal directions as well as per directions. That means in the directions where basically directions towards this particular axis, or like the sidewise basically distributions. So if we consider the aggregation kinetics, initially the molecule shows very slow, you can say accumulations of molecules, and with time like it produce a certain nucleation structure or certain nucleus, and once it forms the specific nucleus, it rapidly progress and produce the amyloid fibrils. So this particular phase called saturation phase, and this one is growth phase. So one can actually look at the structural, as I mentioned earlier, the ensembles in the lag phase and amyloid, particularly the mature phase. So all these phases in a given conditions, you will observe that not every molecule reach to the lag phase, transform to the growth phase, and transfer to the amyloid fibers. In the solution, we observe the diversity of like the species present, like combination of lag phase molecule, growth phase molecule, as well as the amyloid fiber molecule, and all of them behave actually differently towards the neuron. So again, the polymorphism of fiber. So now I talked about the formations of different, like it shows that the periodicity. So this is basically E46K mutants. The periodicity difference, you could observe there the lateral basically the deposition of alpha-synuclein molecules and produce thicker fibers. And like this is cryo-electron microscopic image structure determinations, like recently people have done. You could see that the polymorphism is related to the rod shape polymorphism, where distributions of different monomers is like this fashions, and like the twisted polymorphism is basically this kind of fashion. So within similar, like the wild type alpha-synuclein, not mutant, wild types alpha-synuclein, you have diverse type of the fibrillar structures. Basically, even these two already been identified, but there are large number of reports people have observed varieties of the structural distributions within the fiber. So again, this particular complexity is also contribute to the disease. Yes. No. It is a nature. Mutations cause like different of structures again. Like you some like the bending of these particular structures also some cases observed. But this is naturally like now occurring, or you can say wild type alpha synuclein. It is not mutant. So again, this has like the structural distribution. Here, this paper has shown only 2 different type of fiber morphology. However, large number of fiber morphology people have described, like again, different types of fiber morphology is described in depending upon the conditions given. As earlier mentioned, it is intrinsically disordered proteins. So you imagine like how many it is 140 amino acid like residue. So how many structural ensembles can be formed like based on the Leventhal paradox, if you consider. So many conformations can be basically produced. And indeed, like we observe large number of structural diversity in case of fibers. And each structural diverse fiber shows also different types of biological behavior. Its toxic cells, its membrane interactions, basically you can say to the cells, they behaves differently. No, they are not covalently linked. This is a self-assemble with stacking. But they are very strong hydrophobic interactions. So NAC particularly, it is basically a antiparallel beta sheet structures. It's stacking of beta sheet structures through the strong hydrophobic interactions. And these interactions basically produce like 1 by 1 beta stack structures produce, and then they form this kind of structures. So, yeah, structure as like the general, the process will always, if we consider the thermodynamics principle, process will always drive to the stable structures. So, if we consider stable, so like though the systems is basically like if we consider parameter of thermodynamics, systems always prefer to reach highly intensive, or you can say highly disorder conditions. It means the entropically driven conditions. So that you have more number structures, not this kind of like the rigid structures. May people, one can assume that it will be because it produces large number of entropic contribution. However, hydrophobic interactions, when they are each other, they are restricting. But when they are present, they are in a monomeric conditions, large number of other like the environmental are present in the interface of that particular hydrophobic region. And once they stuck each other or they form this kind of fibrillous of that molecule happens, and overall, like if you can see that overall entropic basic distribution or entropic contributions is enhanced significantly due to that. So ultimately, the delta G associated with this structure negative and stable, and because of that reason, they drive this structure formation. Though like alpha helical structures or like different kinds of like loop or this kind of structures, secondary structures, also some component they present. Like many cases, many the proteins where the intrinsic disorderness is not there. Like we consider the proteins, particularly tau. So tau also, like though initially they present certain kind of disorder structures, but they are not completely disorder. Then the great example is prion protein, PRP protein. So prion proteins, if you see, it is actually rich in initially in rich in alpha helical structures. So due to the mutations or like modification, prion proteins basically initially misfold, and then they subsequently, after misfolding, they enter into the beta sheet structures. They are beta-rich structures. No, it's okay. Like you can. Yes.
In case of this polymorphism for this alpha-synuclein protein, we showed that the alanine gets fitted to threonine or serine kind of amino acids. It seems to be a bit more polar. Use the specificity of the oral structure.
See,
Hydrophobic.
so hydrophobic, this particular, the mutations present in the N-terminal region. So N-terminal region and C-terminal region is unique region. Earlier I mentioned the calcium homeostasis or calcium transport majorly done by C-terminal region. So C-terminal regions carries large number of glutamic acid and aspartic acid. So they are actually negatively charged. N-terminal region actually carries large number of arginine and lysine residue, positively charged. And that allow the membrane binding. So there is a motif, specific motif, like it's called K. So in the diagram, we actually put small boxes. So these boxes, 5 KT KEG motifs are there. So these motifs actually responsible for binding with the membrane. So if you mutate the lysine residue of that particular motif, any one of the motif, you will see impairment of the membrane binding. So the threonine, so very fine balance of membrane binding and the aggregations, as you mentioned, the aggregation kinetics associated with alpha helix and very rapid. Once it bind to the membrane, immediately form alpha helix and rapidly they convert into the beta structures. So the intrinsically disorder, it will not actually disorder to the beta, it actually proceed through the alpha. Okay, so because of that reason, like this particular residue contribute to the formation of alpha helical structures, like the alteration of alpha helical, which delay or accelerate the formation of the like overall kinetics of the particular structures. Yeah. So I discussed earlier the polymorphism associated with structures, beta seed structures. Again, another complexity of this molecule is conformational diversity of oligomers. So earlier there was a debate like which component is more, which component of alpha pathology. So earlier belief was the as it is so the fibrillar structure is the major driver of the pathology. However, later on people find this, it's called oligomers, actually more toxic or more dangerous for the neuronal cell compared to the fibrillar. So fibrillar stage, and it is kinetically controlled and also thermodynamically controlled and reach to the stable conformation. Once it reaches to the stable conformations, they are proteolytic resistance, the environment, like cellular environment, stable. So they cannot be actually bring into the smaller fragment very easily compared to the oligomeric structures. However, oligomeric intermediate, which basically starts in the early stage of the aggregations, what we will say the lag phase of the aggregations. So because of the structural diversity, it can adopt many different types of conformation. And these conformational basically changes actually drive the disease pathology differently. So one of the important aspect, like the people actually study the oligomeric diversity towards the Parkinson disease, like using antibody-based therapy, single-chain antibody-based therapy. So people have initially thought that if we could neutralize this kind of oligomers, we will be able to reduce the alpha oligomers from the brain or tissue or neurons, lead to the curing of these particular diseases. However, because of this oligomeric diversity, here one electron microscopic image are given, but like the current literature, if you see that again more than like the 500 literatures are there where the different type of oligomeric the structures and their cellular functions, biological functions is already been described. So this again, the diversity of oligomeric distributions, it is very difficult to target by antibody therapy. So again, like the electron microscopic image, it shows like it is driven through the kinetically controls and thermodynamically controls interactions. So here recently we actually see earlier, as mentioned, it initially formed the alpha helical structures, produced beta sheet structures with like a rapid conversion. So what this is the work what Monisha has done. Different type of, we wanted to know whether the alpha helical component or beta rich component is actually contribute to the disease basically burden or disease pathology, and like what is their physiological like relevance towards the like different like the biological scenario like neuroinflammation as well as the cellular toxicity. So we could see that the alpha helical structures and beta sheet structures produce different types of shape. Beta rich species produced like this kind of solid round shape species, and helix rich species form the donut shape. And these different species actually contribute different types of like behavior to the neuronal cells. And they also bind the TLR, which is basically one of the receptor for neuroinflammations, for the alpha synucleins, different type of oligomers. So the alpha helical species and beta rich species actually behave the TLR2 or TLR4, which is basically recognized by alpha synuclein molecule. So again, we actually try to find whether the alpha synuclein different types of species. Here, the earlier study we discussed the alpha helical and beta sheet species, how they actually contribute the neuronal toxicity, or you can say we studied their behaviors toward the neuroinflammation. Here in this study, what we try to see the different type of oligomeric forms or oligomeric structures, how they actually contribute to the like the cellular functions alterations. So generally, the alpha synuclein or this kind of molecule, due to the impairment, as I mentioned, like the vesicular trafficking, they actually enter to the synaptic terminal and reach from one to the other. So neuron to neuron transmissions. So these neuron to neuron transmissions happens via different mechanisms. One of the mechanism is basically through the exosomal like the transmission. Another mechanism through like the nanotubes recently been discussed. These extracellular alpha synucleins, basically, if we expose to the different types of neuronal cells, we could see that it forms this kind of like structural features. That means the membrane of that particular the cells basically corrode, or you can say removed from the particular positions and create a small hole. So you could see that like this is basically three-dimensional image. So it is a two-dimensional image. You could see the 3D surface. You could see that different positions more holes are formed. So this is other type of cells like this is again neuronal cells, but this related to the BS we use as a BS as a control. So it is specific to the alpha synuclein. So because it can bind strongly with membrane, so upon membrane it leaches the membrane and create the membrane pore. So and once it produce the membrane coat, it can enter into the cells. Again, cellular entry of alpha synuclein is not necessarily always through the membrane pore. There are receptor also present there. There are again mechanism by which basically they can engulf the alpha synuclein moiety to the cells. So this is pathogenic basically alpha synucleins, pathogenic oligomeric structures. If you expose the cells or like cells exposed, neuronal cell exposed things, they can create. So this is specific to the neuronal cells. Other type cells like HeLa cells or Hex cells, you will not observe basically the formation. So it is associated with the brain which present in the neuronal cells. So this is again like some we have done some analysis by which we could actually prove that yes, these holes are not artifact, these are actually true holes. So in this particular work, we demonstrated. So once like I showed the picture over here in the surface, but how this particular, once they enter into the cells, they actually drive the mechanism associated with the neurodegenerations, like neuronal cell death, neuronal toxicity mediated with caspase activations, along with nitric oxide signaling like alter aberrations, leading to there are modifications of important proteins which lead to the dysfunctions and cause the neurodegeneration. So here the conclusion is basically the different types of oligomeric form. So here I showed the pictures like different oligomeric forms, how they eliminate or the membrane from the neuronal cells and enter into the neurons. So different mutants actually contribute the removal of the neuronal cells, like the surface different entry also, like the quantum of basically protein present in the cells also vary because of their the whole distributions differences. So again, the like the story I like to put here, that like so far we mentioned mutations or the wild type alpha synuclein in spontaneous aggregation condition, it produced large ensembles. However, another complexity which actually produced is post-translational modifications. So again, post-translational modification, if we consider large number of post-translations are there, but some of already been studied in the context of alpha synuclein. So one is basically phosphorylations, acetylation, glycation, truncations, ubiquitinations, and nitrations. So apart from that, like the one can actually study whether alpha synuclein can undergo many other metabolic modifications or not. So there are huge scope in these particular directions. So phosphorylation is one of the important modifications. Like this is already been discussed in the literature many years, almost 20 years, and position C-terminal residue serine 129 in pathogenic conditions like mark of like the Parkinson disease is presence of phosphorylated alpha synuclein accumulations in the Lewy body. So in the Lewy body, we observe large amount of accumulation of phosphorylated 129 like alpha synuclein, which is basically drive the spontaneous aggregate formations. The aggregation of alpha synuclein is basically phosphorylated alpha synuclein is very fast compared to the normal one. And people have studied the synthesized the semi-synthetic like the method, the phosphorylated conditions, and then they studied the aggregation behaviors and many other like biophysical studies. So the ubiquitination is also already been studied. People have demonstrated the N-terminal like lysine residues, like they also modify by ubiquitin moieties, and again that modulate the aggregations behaviors. They produce different types of structural ensembles, similar to the nitrations and like acetylations and glycations. Truncations basically is also observed in clinical like conditions in brain biopsy studies. People have demonstrated the truncations 103. So it is a 140 amino acid residue proteins. So most prevalent truncations is observed 1 to 103. So which is basically the removal of C-terminal region. So once this component is removed, it's spontaneous. The aggregation kinetic is much faster compared to the full-length proteins. So N-terminal is basically somehow restricting the alpha-synuclein spontaneous aggregation. So another modification, as I mentioned, the glycations, which happens due to the metabolic deregulation. So like in the glycolytic pathway, what we observe that like the methylglyoxal is one of the component which produced as a byproduct. And these methylglyoxals plays a very important role in, again, not only in the disease biology, also in physiological conditions. So, and we have systems to remove methylglyoxal. So, one its function is basically finished. So, methylglyoxal modification, lysine residue, arginine residue, and cysteine residue produce different types of structures. So these structures are called like advanced glycations in product. So apart from these methyl glyoxals, from glucose, like if we take a different type of sugar, so there is a rearrangement in the sugar chemistry called Amadori rearrangement. So it produced a active carbonyl compound that also produced the advanced glycation in product and react similarly with residue and product, again related to the lysine residue and arginine residue. Alpha-synuclein didn't have arginine residue, only lysine residues are present there. So it actually modify large number of lysine residues spontaneously. And clinical study, so the study from the brain tissue biopsy demonstrated that like the yes, approximately 5 to 7 lysine residues are modified in the PD conditions compared to the control. So now in vitro, if you consider, if you see, this is the normal aggregation kinetics, it follow the lag phase as well as basically transition phase and stationary phase. However, in these particular modifications, it actually restrict, the aggregation is not proceed. So it form nice fibrillar structures. However, these modifications actually produce like this kind of oligomeric structure. Round shape. However, in case of the modified alpha-synuclein, you will see that this kind of basically rod shape structures and the few molecules are stitch each other. So it is not like only one molecule, many other basically the structures are stitched to each other. So again, the diversity of this species is contributed like that. So another important aspect, as I mentioned, it produced the glycine residues modifications by different types, the methyl glyoxal, once it modified, the lysine is positively charged, it produced the negatively charged because the methyl glyoxals basically hydrolyzed and produced to the carboxylic acid moiety. So N-terminal residue which bind to the membrane, it actually positively charged. Now, due to this modification, it convert into the negatively charged. So if you see again, the electron microscopic image, the large number of like the N-terminal and C-terminal like residues are distributed. The beta stack structures are like this, and you have large number of residues which is basically the projected outward directions, and they are very much prone to modify with this kind of metabolites. So if we measure the zeta potential, the potentials like due to the mutations, what are the charge state in these particular structures, we observe that yes, due to these modifications, it actually produces more negatively charged in the N-terminal domain. And because of that particular modifications, it actually alter the property. And it also inhibit the recruitment of the wild type alpha-synucleins. It also like the primary nucleations and secondary nucleations of the alpha-synuclein molecules and proceed for the fibrillation. So because of that reasons, these molecules never attain fibrillar structures. So now I will not go details like how this particular structure contributes to the biology. So, but we prove that this particular molecule shows completely different behavior compared to the wild type alpha-synuclein in case of its cellular like toxicity as well as the neuroinflammation property. So, like we are like in the process of publishing this work. It is already available in bioarchives. One can look at in this particular the publications. We mentioned like the different types of genetic factors contribute to this, to the PD pathology. One of the important genes called PARK7, Parkinson associated, like it's called Parkinson associated RIX, like the associated gene, which is named 7. Its name is DJ1, which is basically a D-glyoxalase enzyme. So here the methyl glyoxal modification. So once the protein is methyl glyoxal modification happens, this methyl glyoxal moiety, advanced glycation moiety, can be removed by like 3 enzymes in our cellular systems. It's called glyoxalase 1, glyoxalase 2, as well as there is a protein called or like the Park7 is involved in the Parkinson disease, so name is given. So this protein is a metal-dependent chaperones as well as the D-glycan. So its polymorphism is also like the alpha-synuclein. Its polymorphism is associated with the PD pathology. So people have demonstrated that like the oxidation of these particular proteins actually shows the chaperone. Unoxidized conditions, this protein cannot show the chaperone behavior. The oxidation means the cysteine residue present in these particular proteins, they actually shows the chaperone property if they are partially oxidized. If they are fully oxidized, they cannot show the chaperoning property. So, so we are curious to know why this particular protein is shows the oxidative conditions, the chaperone property, and if this particular oxidative oxidized chaperone has impact on the like aggregation inhibitions or like the fiber modeling of the alpha-synuclein, because this particular protein present in the Lewy body. So these are the informations. Mechanism underlying the chaperone mechanism is not explored long time. We thought like try this particular molecule actually shows the chaperone behavior. Again, this is the kinetics. Like one can inhibit the aggregations behaviors by different molecular chaperones in the nucleation phase, then elongation phase, as well as secondary nucleation phase. So, and like any one particular phase, you could actually restrict the fibrillar structure formations. And many cases, many chaperones are there, particularly dependent chaperones. They can also completely disassemble the toxic fibrillar pieces into the monomeric one. However, this molecule DJ1 is a small, around 18 kilodaltons, and it is non-ATPase metal-dependent chaperones. So what we actually initially measure, that we produce the different types of oxidative conditions, where partially oxidized conditions, like we control oxidation, produce the partially structures, and then we measure the nanomechanical property using the force microscopic tools. So what we observe, the high, like fully oxidized condition produced very strong, like you can say strip aggregates, and like it actually produced very less adhesive aggregates. However, partially oxidized condition produced low strip, like the partially oxidized lowest basically its stiffness, and it has so very strong adhesive property. So, and like these are the mechanical parameters which basically shows that it is actually corroborates the adhesion. Based, we try to see whether it actually inhibits nucleation phase. The partially oxidized can inhibits very strong inhibition of alpha-synuclein primary nucleation. That means in the nucleation phase. Also, I am not showing the data here. It can also inhibit the secondary, basically elongation phase. So similarly, it also like we wanted to know whether this partially oxidized condition, because it has very strong adhesive property, like ATP dependent chaperone like HSP70, HSP104, whether they or 90, they actually disassemble fibrillar structure. We wanted to know whether particular structures can inhibit or disassemble the structures or not. So in the literature, small hits, there are no such kind of study. People demonstrated more small hits of proteins can disassemble the fibrillar structures. So here we actually demonstrated if we incubate this particular molecule with partially oxidized, the fibrillar structures with partially oxidized conditions, with time it slowly break down and produced a smaller structure. So we actually proposed the mechanism because of its very strong adhesive property. It wrapped the alpha fibrillar structures, and because of like wrapping, it generates basically the pulling force generate, and because of that reasons, basically it slowly leaches the alpha-synuclein molecule, and subsequently it break down and cut and produces smaller structures. So then we wanted to know what are the fate of this particular structure, whether this structure is important or relevant in the biological scenario or not, because as it is present in the oxidative condition is also present in the Lewy body. So one could presume that with long time basically presence of this kind, if DJ1 partially oxidized conditions are there, they can actually leach this kind of species from the Lewy body and distribute, or basically the propagate to the healthy cells. And we actually demonstrate this species, this kind of species actually shows much higher toxicity to the oligomers. So this is basically the conclusion of that particular story. So now I touch upon the other aspect of, like, so far we have discussed with the mechanism by which basically we showed that alpha-synuclein, the aggregations and behaviors. So as Lewy body is basically one of the complex composite, we observe that, like, the mass spectrometry analysis actually shows large number of proteins are present. Almost 600 proteins are present, and 60 proteins are present, like, as a enriched alpha-synuclein component. So we could see here ubiquitin signaling molecule, oxidative stress protein trafficking, lysosomal degradation, and chaperone molecules. You could observe many proteins what, like, now we already discussed, or they are associated with the present this particular Lewy body composite. But how they actually, and there, how these molecules are sequestered, not even, like, the many proteins already been studied, but many of them is not been studied. So one of the important regulator of Parkinson disease pathology, polymorphism associated with the alpha-synuclein, is one of the highest highest abundant proteins in the brain. So this protein's polymorphism is associated with the, again, Parkinson disease pathology. So similarly, we also observe there is a protein called ubiquitin specific protease. We found that these particular proteins, this basically experiment is not ours. We reanalyzed the mass spec data. We found ubiquitin specific protease O2B1 is one of the important basically deubiquitinating enzymes which regulate the K63 as well as K48 ubiquitin chain homeostasis. This particular molecule is also present there. Despite 100 deubiquitinates, we could find in the T pathology or PD, like Lewy body, the couple of deubiquitinates are present there. So we are curious to know why these O2B1s are present, whether these particular proteins intrinsically inherit certain kind of disorderness, or they actually show certain kind of instability that lead to the aggregations. So when we studied actually that point, like the O2B1's role in neuronal function is not being discussed at all. So, but recently, couple of studies, people have shown that it has also play important roles in tau pathology. So, ubiquitin systems, I don't have to discuss that much. Many of you already aware of these things. It actually involves ATP dependent process involved with E1 enzymes, E2 enzymes, and E3 enzymes. E3 is basically select the substrate and ubiquitinate, and E2 is basically conjugating enzymes which transfer the ubiquitin from E1 to E3. And subsequently, it actually drive the different types of signaling. Like one is basically proteasomal degradation, lysosomal repair, on many like the biological process it can regulate. So the Parkin is one of the important regulatory genes which is basically responsible for PD, and these genes also important role in mitochondrial functions. So Pink and Parkin, they combine together and actually regulate the mitochondrial genesis. So this is also important, like ubiquitin ligase, which regulates the Parkinson disease pathology. Another is basically usagelon, as I discussed. So USP8, which basically indirectly regulates the proteins basically with alpha-synuclein loads in the brain. However, O2B1 was not known, so that's why we thought like whether we could actually study the O2B1 functions in the context of basically the Parkinson disease. So like again, like here it has two types of function, canonical functions. It is a unique DABs which basically contain canonical functions and non-canonical function. Canonical means it is a DABs activity. Non-canonical means it actually interacting with other regulators. It regulates the functions of ubiquitination and deubiquitination. So, so this is the one deubiquitinate enzymes. It regulates both canonical and non-canonical functions. So it's functions of cancer, DNA repair, and immunity is very much well established. However, neurodegeneration is not been yet discovered. So it interacts with so many ubiquitin ligases. Ubiquitin conjugating enzyme, human genome encodes approximately 27 ubiquitin conjugating enzymes, and it actually interacts with approximately 7 to 8 ubiquitin conjugating enzymes and regulate different types of recruitment, or you can say different types of cellular functions. So initially, actually curious to know why this particular proteins is present in the Lewy body, whether these proteins actually carries the in itself instability because of that present there or not. So O2B1 is basically again alpha beta proteins, it majorly alpha helical, but it also possesses approximately 25%, actually 22% of beta C structure and 45% of the alpha helical structures. So we actually wanted to know in vitro conditions whether this molecule actually shows the amyloid behaviors like the O2B1s or many other like A beta. So we started in a biophysical, these are all biophysical methods. We demonstrated, yes, it actually shows very strong THT dye binding, which is basically a dye detect the amyloid structures. Again, it specifically interact with the antibody specific to the fiber as well as oligomer. It actually shows the cross-polarized light, the green represents the basically again amyloid structure. Again, conversion of alpha to beta, this conversion we could observe using the CD spectroscopy, and using the atomic force microscopy single fiber analysis, we could see that there is a distinct periodicity present in the fiber. And this is basically time dependent, like the accumulations, the transition kinetics. If we see that there is a formations of different types of oligomeric intermediate. So in this particular, we demonstrated that yes, this particular molecules basically plays an important role in the neuronal toxicity and in environmental exposure condition, mitochondrial dysregulated conditions. This molecule also produce the spontaneous aggregation in cells. And in animal model, particularly in PD mice model, we demonstrated these particular proteins also produce the increased clean phosphorylated form and co-enriched, or you can say co-localized with the alpha-synuclein S129 pathogenic form. So so this is all about. So from that, we actually demonstrated it produced the cytotoxicity and lead to the Parkinson disease pathology. So this is all about. Now I will stop here, and then if you have any questions, then I will be happy to basically address them. Yes.
Parkinson is more prevalent in males because the ratio is 3. So is there any gender related reason known that males have a certain hormones because of which?
Generally there are the female hormones, particularly they are protective in their early age. So juvenile Parkinsonism. So if you see the Parkinson is again classified in a generally people observe the Parkinson disease above 50. So once it consider that there is a movement disorder society, there is a scale, specific scale where they describe that many symptoms appear after age of 50 to 70 or old age. This considered as basically Parkinson disease related to the normal normal conditions. However, if it is less than 40, so then we consider or even very early age, 15, 20 or 25. So it's called juvenile Parkinsonism. So majority of juvenile Parkinsonism is observed is largely the male. So again, in menopause, post-menopause, the women are very much basically exposed. If they are exposed to the different environmental conditions, they actually show this Parkinsonism. However, the recent study, our group, we are studying the contributions of you can say the diabetic conditions. If we observe that diabetes actually shows much more risks to the women for Parkinsonism compared to the male. Okay, so that like this is specifically we are doing the animal study. However, recent like 2021 one like human study people have demonstrated the women's are almost 30% more, or like the more you can say prone to the Parkinson disease conditions if they are like exposed, they are actually exposed to this kind of metabolic disorder, particularly the diabetes.
In case of this additional glycation end products, you said that it's shown that this glucose leads to glucose kind of molecule carbohydrates leads to additional glycation products, and they're promoting the aggregation formation. So can it be said that the fat-based diet, okay, where you have more of formation of ketone bodies, okay, which are far more digested part, okay, acetyl CoA and all those, so they would be a bit better for those who have chances of making the developing Parkinson's in their older age?
Actually, yes, that could be, but again, there is no such kind of experimental evidence whether this kind of like diet actually prevent the AG related Parkinsonism or not. Like this has not been studied. However, there is a scope like one can actually look that whether the ketone body is related related to like the Parkinson disease, is there any correlation or not? But like yes, this has not been studied. So yeah.
So in this monomeric form of alpha-synuclein is very simple, but fibrils, generated diverse, structures. So is there any suppressor mutations of these polymorphism that reverse their effect, or can suppressor mutations reverse because these are the fibrils?
Suppressor mutations, like so far, nobody has already like reported, or people didn't find. Large number of genome-wide association study has been carried out. But people didn't get any suppression mutations related to the alpha-synuclein. Again, as you mentioned, the good mutations in case of UCH, again, this is also one of the genetic factors. UCHL1, there is a mutation called I93M. So this mutant is basically responsible for like called S18Y. The S2Y, these mutations actually protect. The study already been like discussed like with some population studies, but in vitro, like if we see the impact of these two mutants, S18Y as well as I93M, I have done this study earlier. I could not find any difference structurally as well as their like thermal behavior, stability, interactions, I could not find. So could be something when they are present in complex systems. But the structurally, those both mutants are very much similar. Stability-wise, both mutants are very similar. But they are like described as the protective mutants because they found like these mutants present there, that persons are.
So these two cysteine in the DJ1, whether they make a disulfide bond, if so, then why this partial is giving that? Okay.
Require a certain distance, so they are apart from like I think not within the covalent bond formation, they are away from the disulfide bond formation. But again, the cysteine residue 106, age group. So it is basically one OH group will be attached. So these particular age modifications actually structure somehow secondary structure, if you consider secondary structure somehow actually alter, but it is not completely disrupt. However, of these disrupted structures, they form like again oligomeric, some kind of assemble, and that actually wraps. So chaperone literature people have discussed like many, I think 10-15 yes, the condition what we made, only that condition. Molecule. That's basically we are curious to know. It was not known, so we thought like whether
There are no other questions.
Sir, these Lewy body and the fibrils, they are present in the cell. How they are avoiding the defense system? Why the defense system is not acting against them when they are not like?
Yes, defense systems actually act on them initially because this process is not very quick. It is actually a slow progressive process. It start Parkinson disease. I showed the number 100, 200 numbers in 1 lakhs. Compared to other many diseases, these numbers are very small, but it actually contribute huge burden. So points that progresses very slowly. Initially, our like the proteasomal system, lysosomal systems, they actually try to remove. Certainly they remove certain part, they remove. The protein overload or proteostatic overload, initially cell will try to clear. And once they produce the fibrillar structures, they, as earlier mentioned, is a proteolytic resistance because they form, protease has to enter between this fibrillar structure and cleave. So actually enter. So these molecules are very strong, even in they are insoluble in SDS and like the denaturant. So very strong basically structure. So with like the structures, because of that reason, like the system, particularly proteasomal as well as lysosomal clearance systems, and again the clearance by microglial engulfing these molecules, they actually failed. So it not initially failed, like with time it will progress and subsequently like the pathology will appear. So pathology will not show the progression of disease, like initially it starts like the bradykinesia, tremor, this kind of the component is ultimate manifestation. But it start like one of the difficult actually diagnosis of Parkinson disease is basically the group where you have this kind of syndrome, you can easily segregate. Large number of Parkinsonism is there where you do not have like this kind of tremors are there, you do not have like the other like the problems are there. So they also shows like the impairment of like you can say Lewy body pathology with dementia. You do not have or something, but Lewy body present and you actually enter into dementia. Dementia means you are basically forgetting like all this. So because of that is in like initial part you will see, but slowly with time other basically manifestation will come. So one of the important or you can say difficult part of the clinical basically study or clinical the Parkinson disease study is characterizing different types of phenotype, different type of disease basically outcome. So there are conditions called rigidity, like the dominant, tremor dominant, as well as like the echinacea, bradykinesia dominant, and like there are called like atypical Parkinsonism, which is basically like as I mentioned, mostly it is related to like those who are taking different large amount of drugs. Almost every day people are taking different type of drugs for different disease conditions. So that drug also actually induced Parkinsonism. So many cases you will observe that like drug induced Parkinsonism is also observed. You will not find the symptoms as the classical symptoms. So these are called atypical Parkinsonism. So that also actually happens. So major challenge is basically like cell after certain point fails and cause these kind of like behaviors.
Sir, I have a basic data. So I want to know about the basic data. So what is the basic function of this alpha-synuclein apart from the role in pathogenesis of this PD?
Yeah, so I earlier mentioned
Really
like
the
actually
monomer.
these particular proteins like basic trafficking process, particularly the neurotransmitter release, neurotransmitter transfer from like of vesicle. So this particular vesicle, the alpha-synuclein actually embedded in the vesicular structure and allow the formation the normal functions. So like these people have observed that functions, but there are many functions could be physiologically relevant, but actually complex like the snare complex, snare 25, these complex vesicle like the neurotransmitter packed with this particular complex. So normal alpha-synuclein plays a role like without disease condition, but gene duplication, if you have more number of alpha-synuclein concentrations due to the production of more alpha-synuclein or like different mutations, the vesicle structures basically alter and the alpha-synuclein released from that and packed into the, you can say, exosomes, or like it can transfer from one cell to the other cell through other different mechanisms, like nanotube is one of them recently been appreciated, so they can transfer. So this the normal functions, but again, like if you Google it, you will find this is not like the function of alpha-synuclein is not explained or not known very well. Yes.
If there are no other questions, let's thank our speaker, Dr. Tushar, for a wonderful talk.